want to show you how using a genetic test like the Tree of Life can really help you identify some of your root causes of your problems. So here is a pyramid map. And I just kind of scan through some of these things that I see that could be problematic. So for example, I take a look at the fencing reaction to see if there's any iron absorption or any potential for reactive oxygen species. And then I go into NOS uncoupling, and I can see here that this person has a homozygous polymorphism in NOS2, which can be upregulated, which it is, which is going to drive up reactive oxygen species. And they also have a downregulation in NOS3 that's used to quench that process. So that's one area there. Then I jump into food, gut, and histamine oxalates, and I can see there's a couple of histamine pathways here activated. We've got the histamine breakdown. We've got the DAO enzyme. Nothing on the oxalates, but I do see some people that do have oxalate genetics. And then I will jump into the same thing here to look for the hemoxygenase 1 and 2. There's some heterozygous polymorphisms there that could be problematic. This is your mast cell. Nope, nothing there, so that's good. And then I go into glutathione to see if there's anything. So that we've got some transsulfuration potentially, definitely glutathione recycling, another antioxidant pathway. And then we've got the NRF2. This is key with IC. People who have the chronic embedded infections have polymorphisms, and they tend to have the NRF2 with the KEAP1 upregulation. This is one copy, so heterozygous so definitely can be contributing to the biofilms and the embedded infections. Then I took a look at SOD, superoxide dismutase and catalase, more antioxidant enzymes. We do have a downregulation there, so that's not good. And then I can jump into sulfation because sulfur is so important for oxalates, and this person definitely has a few here. We've got SUOX and we've got the salts, our enzymes in sulfation, and it's another phase 2 detoxification. We have glucuronidation. There's a few here, nothing crazy, but enough to say there could be some issues with glucuronidation. And then finally methylation here. We've got 3 homozygous in COMT, and that can be very, very problematic when it comes to detoxification of things such as estrogens. And then there's your methylation upregulation. So this person would probably not be a very good candidate for vitamin B6 therapy. And there's your methylation pathway here. We've got, 1 homozygous. That's pretty significant because that can affect homocysteine and methionine recycling and availability of certain nutrients. We've got the PEMT, so definitely needs some support with methylation, potentially having, looks like there may be some B12 absorption issues here. And so what I do is I pair this up with a nutri-eval to see which of these genetics could actually be activated or could be expressing. And finally, there's PON1. That's your glyphosate detoxification, and this person could have some issues with removing glyphosate out of the body, which is also very disruptive of the immune system, but can also be negatively impacting your, can also be contributing to oxalates.